All right. I am working on a class project for Pedro Mira's course. We've been doing a lot of RNA-seq, kind of next generation sequencing work and learning different pipelines and methods for analyzing next-gen data and primarily RNA sequence data. So for part of my project, kind of compiling or putting together a series of little tutorial videos, although my camera's not working, so hopefully my screen share will. But I want to kind of put together a series of videos that take you through finding maybe sequence data through the NCBI database, learning how to use the SRA toolkit, which is the Sequence Read Archive toolkit, to upload and procure that data to your local computer, and then working through HiSat pipeline to work with those sequence data, and eventually, hopefully, we'll get through some differential expression analysis with that. So this portion, or this video, I wanted to just work through kind of how I found data on the Sequence Read Archive, and then how I used SRA Toolkit, which is a toolkit that you can download to use to upload that data. So to start out, I'll just come here to the NCBI website. So if we come from the home page, we click on the Databases tab and find the SRA. This is the Sequence Read Archive. Then you can type in some keywords. So what I typed in was neotoma. I work with wild mammals in the genus Neotoma. These are wood rats. And I wanted to know, do we have any RNA-seq data? And so what I found here, yes, we do have some data. And what I found were these three in the middle here are from a single experiment and paper that was published by Kevin Cole. I've got that paper loaded here. Okay, so it's this paper here, Patterns of Host Gene Expression Associated with Harboring a Foregut Microbial Community. So in this paper, in short, they were interested in finding patterns of gene expression in liver tissue in a wild wood rat, in this case the Stevens wood rat, Neotoma stevensi, and they compared that to a lab rat. So the Stevens wood rat harbors a dense foregut microbial community, and the lab rat lacks such a community. So they're interested in determining if there are any patterns then in differential gene expression associated with herbivores with two different kind of foreguts in this experiment. So if you read through the paper, and this is how you'll oftentimes find how to access data, but if you look at their methods, at the very end, they say all reads have been uploaded to the NCBI Gene Expression Omnibus under a session number, and they list it out here. So this is the accession number they give for the GEO, or the Gene Expression Omnibus database, which is similar to the SRA or Sequence Read Archive database. I have just used the SRA data for this example, but just know that whether you're using GEO or SRA, it should be fairly similar. And I think, well, at least in the case of this paper, they report that they archived their sequences into the GEO database, but they also exist in the SRA database. So if we just look here, we have 3 replicates of Neotoma stevensii, and in this experiment, they also had 3 replicates of Rattus norvegicus, the lab rat. But if we just click on one of these and open it, we'll see that there's a different information. We have the library prep and kind of metadata in terms of how these data were prepared, what instrument was used to for the sequencing. And we also see these different project numbers and experiment numbers. So if we click on all experiments, we'll actually see every all the sequences for each of their replicates. So they have 3 replicates of the rat and 3 replicates of the wood rat here. Okay. Now, what we want to do, if we want to download or obtain this information onto our own computer, we need to use the SRA toolkit. Which is downloadable. If we just look at SRA toolkit, we can download the correct version of this program depending on what operating system you use. So I have a Mac, so I download this version. Once it's downloaded, you can use it. So if we go back here, we want to download each of these sequences. Now, to use the SRA toolkit, we need to find the correct identifier number for each of these sequences. So if I click on, let's go to the replicate number 1 for Neotoma stevensii. Real quick, I'm going to quit that so it's not. Excuse me. Okay. So what you're going to find are these different identifier numbers. This is the bio project, so a link to the project. This is a link to each experiment. This is a link to the SRA study. But what we really want is this run number. So this is a code that starts with the 3 letters SRR and then followed by several digits. This is going to be what we actually use in the SRA toolkit from the command line to get the data. So if I click on this, we can actually look at some of the metadata. We can look at the quality graph. We can look at the experiment. We can look at the biosample. We get a little information on the project again. So you'll notice if we look at some of these different tabs, we can get an analysis. We get some information on the reads. But then to download, you can't just simply download it into your computer. So another link to this SRA toolkit. Okay. Now I've already downloaded this toolkit and have the program operable on my computer, so I don't need to do that, and I won't do that for now. But I will show you kind of what I'm working with. So I'm on my computer here, and I'm actually SSH'd into another computer in my lab that's got more computing power. But if I just look at my directory, okay, I'm in the correct directory, I'm going to list out that directory. I've already worked through several of the steps before making this video, but what we want to get from the SRA toolkit using SRA toolkit commands are these fastq files. So you can see that I've already generated fastq files for all three of the wood rat samples, and I've actually also downloaded the fastq files for one of the Radis sequences. So to show you how it works, I will download and generate another fastq file for one of the other Radis norvegicus sequences here. So before I do that, I just want to show you kind of this SRA toolkit. These are notes that I've been keeping. So the first command or kind of function we need to use is this prefetch. Prefetch is simply going to kind of create a connection to the database for that particular data set given by this code that starts with SRR. Again, that will be this number here. So these SRR followed by numbers, these are actual runs, each associated with an individual replicate or sample from this experiment. So prefetch is a pretty simple command. All you're going to do is type in prefetch, and then we want to give it the correct code. So I wanted to download another one of those Radis norvegicus sequences. So I'm going to take replicate 2. So if I click on that, I can obtain this SRR number that I need. So it is SRR 3902566. 3902566. Now, it should run. Yeah, it looks like it's working, so it's downloading. So the nice thing about this toolkit is that unlike from my experience using FTP to download or access data, you have to give it a path or like a web address for where to obtain that information. But because this SRA toolkit is built specifically for Sequence Read Archive database data, it's already kind of got the path built into the algorithm. So you just need to give it that SRR identifier code, and it will automatically download the correct information. So again, pretty simple. While that's running, I will show you the next step is going to use this fastq dump command. Now, the difference here is that, or what prefetch is doing is just kind of obtaining the information, but we actually want fastq files. We don't need all the other stuff. So what fastq dump is going to do is, given certain options and parameters that we provide in the command, it will generate our fastq files for our use. I suppose another thing that's going to be important for that fastq dump command is going to be knowing a little bit about our data. So these are paired end sequences, and the read lengths are 100 base pairs. So we have 200 total bases read, and they're paired end. So 100 reads on read 1, 100 on read 2. Or no, actually they're paired in and overlapping, so they should have just 100 reads. So there are 2 reads per spot. It takes a little bit of time. Okay, so we have downloaded successfully that replicate for one of the Radis replicates, so the sequences for that. So now if I look into my directory that I'm in, I will find that. It's SRA 3902566, and we should see that. Oh, we don't have it yet, okay, because all we did was prefetch. Now, for the fastq dump. So I'm going to show you this command here. So it's fastq dump with some options. Now, remember I told you that it was paired end data. So we're going to specify that with this value here, capital I. And then we also want to split the files. Because it's paired in, we have 2 reads. So we want to separate it by read 1 and read 2. So we're going to tell it to split files, and then we're going to give it that same accession number. So we'll run it just like that. I will fastq dump capital I split files. All right, make sure you don't have any spelling errors, and then SRR 3902566. Now this should run, and it takes a little time, it should run and generate a fastq or 2 fastq files for this particular run. And we'll see it eventually displayed like this. So read 1 and read 2 for that particular sample run. Okay, so I'm running that in the background now because I want to show you some of this other stuff. So that essentially gets you from finding a data set on the SRA database, Sequence Read Archive. And again, you can do that just simply by going to the main NCBI webpage, look through the SRA database, and you can use search terms, RNA-seq. Okay, so we did that. Now you could search for any number of things. And again, what you want is the SRR accession number for that particular sample, which is going to be found here for the run number. Once you have that, you use the SRR toolkit, prefetch, and then fastq dump. Oh, it looks like it's maybe finishing up here. So it's done. Okay, well, so there we go. We've created our fastq files for the read 1 and read 2 for that run number, that Radis norvegicus replicate. So I'll go ahead and look at that. Well, let's see. Let's get the file size for that, just to see. It should be fairly large. If not, then you'll know that it's 3.4 gigabytes. So that's kind of what we want to see. There's no issues there. Another thing you might do is use fastqc. So this is a program that you can download. Fastqc. Actually, I don't do it here because the files I have been generated are actually on a different computer. But I can show you this. fastqc is a quality control report program for working with this kind of data. So what you do is you just click on open a file, and then you give it a file, and then it will provide you a bunch of quality stats for your sequences. Too bad I'm not on the other computer because I could actually show that. But if we want to now align these two reads using HiSat, we can do that now that we've got our two reads. So what I kind of want to do is something that I realized here in doing this is so in this experiment they were using replicates of a wild species of wood rat, comparing liver gene expression of those individuals to 3 replicates of lab rat individuals to determine if there were differences in gene expression among different mammal herbivores that harbor different types of foregut microbial communities. So the lab rat is a model organism, and it has a very good reference genome available for that. The wood rat, however, is a wild organism, and there's no real reference genome, or at least not a well-studied genome for pretty much any wild organism. So for HiSat, we have to align our sequences to a reference genome. And so what I found when I did the alignments using HiSat is that the alignment rate for the wood rat, the Neotoma stevensii, was about 9.5%, while the alignment rate for the rat sequences in this study were over 90%. So that's not surprising. But what that kind of led me to do is, in using the HiSat alignment, there are options for how strict you want your alignment to perform. And so this MP option is something you can use to adjust the penalties given for mismatches. So the inputs for MP are a maximum penalty and a minimum penalty. And the default, I believe, is a maximum penalty of 6 and a minimum penalty of 2. So I reduced the maximum penalty to 4 here. This is what I ran for the woodrat sequence alignment. And that increased my alignment rate to about 16%. Now, whether or not those alignments, that increase in alignment rate is real, we don't know. But if you're concerned about the what your alignment rate is, you can adjust some of those options. And so I did that, and actually I don't have to run it to show you. If I look at my history, I should be able to find it. So bear with me while I scroll through a lot of information here. Well, maybe it won't. Okay, well. So I will just do this. I will actually run another one. I will run this. I'm just going to cut and paste it in since I already have the files. So what I have here is I'm calling the HiSat2 tool, giving it 4 processors, setting this is an optional setting here, but I'm setting the stringency, so the maximum penalty is 4. I have the reference genome. That's something that gets downloaded along with the files. So that's another mention I didn't mention. So when we download the all those information, remember we need to align to this Radis genome. So I downloaded the Radis genome through I believe NCBI rat genome. Or no, it was the University of California, Santa Cruz, UCSC genome browser. Is that right? Let's see. Where did I get that? I think it was through the NCBI. Let's see. All right, I'm going to find this. There we go. Okay. Okay. So if we look at the, it's actually in this HiSat2 kind of tutorial. We can look at the right column here, the kind of the right panel, and we have a bunch of indexes. These are all indexes for different, mostly, well, model organisms. But I wanted the rat genome, so I can download the UCSC version. It's the 6th, I guess, edition of this genome. So I click on that. It will download. So I don't want to actually download because I've done that. But you'll download a zip file, and then from that you can store it into the working directory that you need it in, and then you can look inside of that. So if I look inside of that, we actually have different genome files. So this is essentially the index you're going to use, or that HiSat will use for some of the alignment and other steps involved in this. So you're going to need to have that file downloaded for whatever reference genome you want to use for your alignments. But when we call that, we're going to use the RN6 genome, so that's going to be the path for the reference genome we're using in the HiSat alignment. So if I call this HiSat2, we're going to run 4 processors. RN6-genome will be our reference. SRR, and then we're going to do, I'll show you. If I include this optional relaxing the strictness of the mismatches, so I forgot to do that. So I'll give it that real quick. Four, 2, okay. And I will give it one of the files. So because the rat sequence data in this file already aligns pretty well to the rat genome, I'm going to do another one of the wood rat sequences aligned. So I will give it 39025664. So that's read 1 there, fastq file, and then read 2 is going to be the other one, 39025664-2 fastq, and then the output SAM file will be SRR 390264. That's just telling it what we want to call the SAM file that will be generated. Now this should run if I've done it all correctly. Let's see. I think I added. Oh, yeah. Okay. So failed to. All right. Dashes. Okay. Yeah, I just forgot the dashes here, so I need to include that there. Any of these options you need. So I think what it is I just forgot these two dashes. So that's going to take a little while, and I'll just kind of let it run. So at this, so the MP option here is the mismatch penalty. So you get a maximum and minimum penalties for mismatch. So the lower the quality, the lower the penalty. Um, so if you reduce that maximum penalty, um, you're going to it's going to penalize mismatches less the lower this number is. So since I'm working with sequences from a wild wood rat, but I'm aligning to a reference genome that's a lab rat, I know there's probably going to be a lot of differences. So I'm just kind of relaxing that stringency to see if it aligns a little more. I'll still, once I work through some of the other workflow, we'll actually be identifying genes. So we'll know, we'll be able to kind of use our common sense to determine if genes we're finding make sense. Because we know that this is liver tissue, we should be seeing genes that have functions that would be involved in liver function. Okay.